Hello everybody and welcome to today's QCE Psychology Units 3 and 4 January Head Start lecture. My name is Hannah and I'll be your presenter for today. So this free lecture, which is one of 4 that we offer at various points throughout the year, are just one of the free resources that ATAR Notes offers to help students do the best that they can in their studies. We have a whole bunch of other resources available on our website, which you can see on the screen. We have articles, newsletters, we do videos, we have discussion forums and all sorts of other resources, as well as, like I mentioned before, the other free resources and lectures that we do. And then we also have some paid resources such as our study notes. The study notes are really awesome books because they have such concise content summaries of all of the content that you go through in the senior subjects. And you can purchase paper copies of them, but we also have digital copies available on ATAR Notes Plus, which for a monthly subscription fee means you can access all of our really awesome ATAR Notes resources, such as some of our new flashcards that we've got coming out, as well as all of the text that I mentioned before. And there's also a really cool AI answer bot on there as well, which you can ask questions and interactively get feedback on your study with, which is really cool as well. So, like I mentioned earlier, this is our QCE Units 3 and 4 Psychology lecture. The slides are available for download from the resources tab below if that's something you'd find helpful or if you'd like to come back and review them later. And you can also rewatch this lecture video from this very page if you'd like to come back to it as well. And there's also a live Q&A in the chat box down below or off to the side, so feel free to ask any questions in that throughout the lecture today if you have any questions at all, and I'll be available to answer them. So, a little bit more about me. My name's Hannah. I'm a biology, psychology, and general maths tutor at TuteSmart, and I'm involved in some of the resources and lectures as well, like you might have seen. I graduated high school in 2021, and I actually got 97 in psychology, for which I got the Subject Achievement Commendation Award, which meant that I got the top score in the state of Queensland for that year. So that was a very exciting achievement. And I'm also currently studying primary and secondary education, and my major's biology. So I quite enjoy that I get to do a little bit of teaching for work, which I find really, really cool. And now we'll have a look at a little bit more of what we're actually going to be going over today. So today is a Head Start lecture. It's designed to give you a head start for Year 12. I'm aware that a lot of you will have already started some Year 12 content at school, so we'll probably focus less so on the first part of Unit 3 and more so focus on the second part, but we'll still go through quite a lot of the Unit 3 content. We'll get through as much as we possibly can. So we'll have a little bit of a look at localization of function in the brain, visual perception, memory, learning, and then if we get some extra time, we'll go through some scientific skills as well. And just a quick note, we won't actually be able to cover all of the content in today's lecture because it is only a 2-hour slot. So make sure you keep referring back to the syllabus to get the whole scope of what it is that you're expected to know, because if it's on the syllabus, it could be assessed in both your internal assessment or on your external exam. So I've linked that in the slides. So that's something really valuable that you can have a look at as well. And again, just a quick reminder that you can download these slides from the resources tab. So the first thing that we're going to be looking at under the topic of the function in the brain, localization of function, that first topic in Unit 3 is actually the divisions of the nervous system. So kind of getting an idea of the anatomy and the structure so that we can better understand the function and how it works. So the nervous system in the body is all about decisions and communications. The nervous system is one of the systems in the body that really runs our body. It receives information from the external environment, it processes that information, and then it sends out a decision. And as we can see in this diagram here, the nervous system actually has 2 subdivisions, the peripheral nervous system and the central nervous system. So the central nervous system is comprised of the brain and the spinal cord, and it's mainly involved in decision making, and it controls the body by receiving information from the peripheral nervous system, processing that information and sending back a decision. The brain integrates information and makes decisions, and it does that for both voluntary and involuntary functions. So for example, voluntary, like choosing to move a limb, but also involuntary decisions, such as whether to speed up or slow down your heart rate, something that we don't actually consciously control, but our nervous system still has a large role in controlling. And then the spinal cord is also involved in both decisions and communications. So for example, sensory information that's collected from like touch, for example, in your fingers. It might be collected in your fingers, sent to the spinal cord, and then that information is sent to the brain. And then if the brain wants to make a decision with that information, the brain will send that information or that message back by the spinal cord and then back out to the hand. So you can see how it's involved in that like communication between the body and the brain. But the spinal cord can actually also make decisions by itself, which is what's called the spinal reflex, which is where the spinal cord chooses to make that decision of its own accord. Another really important clarification that I actually see a lot of mistakes about is the difference between the spine and the spinal cord. So if you're asked about the divisions of the central nervous system, it's really important that you say the brain and the spinal cord, because the spine is actually incorrect because that's the bone that encompasses it. So just like the skull covers and protects the brain, the spine is like the bone column that actually protects the spinal cord, which is that bundle of nerves that's a part of the central nervous system that's involved in that decisions and communications. So like we said before, the spinal cord passes sensory information between the peripheral nervous system and the brain. It also makes those quick decisions and sends directives around the body. So again, coming back to that idea of communication and decisions. And then again, the idea that the spinal reflexes are those quick decisions that it's really easy for the spinal cord to make. For example, like a dangerous situation, like if I put my hand on like a hot plate by accident, my spinal cord doesn't even need to consult the brain first. It just knows automatically that I need to take my hand back because that's dangerous, and it's actually important for my safety that I really, really quickly make that decision. And then what actually is the peripheral nervous system? Well, that is everything else. So all of the nerves in our arms and legs, everything a part of the nervous system that's not the brain and spinal cord. So that's all over our body, sending those messages, receiving that information and all of that. And it actually has multiple subdivisions. So we have the autonomic and the somatic nervous system. So first, the autonomic nervous system. That's the nervous system that interacts with more of our internal body, such as like our organs. So receiving like stimuli and information from our organs, and also sending back messages to our organs. And then the somatic nervous system, which is more under voluntary control. It's more about like the interactions with the external environment and more about like our muscles, for example. So me consciously choosing to move my hand, that's more of like the somatic nervous system, because it's like my muscles are involved in it. It's about my interactions with the external environment, and it's under voluntary control. And like we covered before, it's still about that communication between the central nervous system and the body. And then the somatic nervous system communicates between the central nervous system and the voluntary conscious functions. So like what we just covered there. So for example, kicking a ball, moving your arm, all of that, interacting with the external environment. And then our autonomic nervous system, so that system that more interacts with the internal environment and controls those like automatic functions such as your heartbeat, that can also be further broken down into the sympathetic and parasympathetic divisions. So the sympathetic is mainly the emergency system that's activated when we feel threatened. So when you hear sympathetic nervous system, we're thinking fight or flight. Whereas our parasympathetic nervous system is our rest and digest. That's our normal day-to-day system that facilitates bodily functions like digestion. It also can contribute to emergency responses such as like the freeze response, but it's mainly thought of as a non-emergency system. So sympathetic, think fight or flight, and parasympathetic, think rest and digest. And this here is just a little bit of like a visual of how the nervous system is broken down. So for example, the central nervous system, again, we can see is like in yellow here, it's like the brain and the spinal cord, and then the peripheral nervous system branches all over the body and is responsible for all of those other areas. Another example, just to illustrate here, we have an example of a dog that's in the snow, and we are going to having just like a quick think about what are the different roles of, for example, the autonomic and the somatic nervous system in this scenario. So for example, this dog's autonomic nervous system would be responsible for controlling its involuntary functions. So it's in the snow, but all of the regular functions are still happening. So for example, a dog's heartbeat is still being controlled by the autonomic nervous system. And the somatic system, which what we talked about was that voluntary control, that interaction with the external environment, that like more voluntary movement would, for example, be controlling the fact that the dog is actually choosing to walk. So that's like a semi-conscious movement of moving those voluntary muscles and again interacting with the environment in that way. So that's just another illustration of the difference between the autonomic and the somatic nervous systems. So we have another example here of the difference between the sympathetic and the parasympathetic nervous system. So for example, sympathetic nervous system, again, that is our fight or flight system. It's our emergency system. So what that does is when that's activated, it speeds up the heart rate, it ensures that more oxygen can be delivered to the body and the tissues. There's more glucose available for the body to use. It's all about mobilizing the body for action, and it's often in the presence of danger. So for example, here we have some very serious like athletes like riding their bikes. They're in like a high energy demands physical situation. So it makes sense that they're going to need an increased heart rate so they can deliver more oxygen, get more glucose so that their body can be ready for action. Another example that I think of with like a sympathetic nervous system is like when you're in a sense of like danger or you're being threatened. So if it's like a bear or a dog or something, that nervous system is going to be very like active because it's about mobilizing your body to act. Your body needs to be ready to run, which is where these changes come in really, really helpful. And then our parasympathetic nervous system is our rest and digest, our everyday system, bodily functions like digestion, like respiratory rate when it's just like regular, all of those things that just need to happen even when we're not in emergency situations. So for example, even just relaxing after a big lunch on the beach. So here's another illustration of like how our nervous system is broken up. So again, bringing it back to that main idea that the nervous system is all about decisions and communications. So receptors detect stimuli. So that could be something from the internal or the external environment. Stimuli is that information that we're detecting that helps inform the decision that our body is going to make. And then our sensory neurons communicate this information to the central nervous system. So it takes it to the brain, it takes it first to the spine, which then communicates it to the brain, unless it's a really easy, quick spinal reflex decision that it can make, of in which case the spinal cord can actually make that decision itself. And then once, I'll just pull up my laser pointer here. So we have our stimulus. Sensory neurons communicate that information to the CNS, the central nervous system. And then our motor neurons actually communicate like the directive, the decision, or the message about what are we actually going to do in response to this stimulus. And that's communicated to the effectors, which is normally like think muscles, glands, organs, the parts of our body that are actually going to help bring about a response. So whereas, for example, in a reflex arc, we actually only have this pathway. And then as the interneurons in the spinal cord are making the decision, so like, right, you're in danger, you need to take your hand off this hot plate, it sends that decision to the effectors. It's like saying, I know this is an easy decision, so I'm going to make that to save time so that it can help the organism survive. And then it also sends that information to the brain. So by the time I've already removed my hand from the hot plate, I've already made the decision to keep myself safe. Whereas in regular like neuronal communication, we do have that information coming, and then it goes from the spinal cord to the brain. The brain makes the decision because that's where we do all of our thinking and our higher order like processes. And obviously the brain is quite specialized in a lot of regions. And then once the brain makes that decision, it sends it to the interneurons, and then from there communicates it to the effectors. So the brain, quite a complicated place. There are quite a lot of different parts, and it's really important that you keep referring to the syllabus to find out what parts you specifically need to know about and in relation to what, because there's quite a lot to know, and if it's on the syllabus, it is fair game for your external. You've quite likely gone over a bit of this in class already, so we're going to skip over this a little bit for now, but there is just a few parts I will quickly draw your attention to. And the first one is actually the cerebrum, which is this big outer part of the brain that kind of like looks like folded sausages. It's also important that you don't confuse that with the cerebellum, which is kind of like that little mini brain down the bottom that's involved in like movement and balance and coordination and like coordinating smooth movements and all of that. But for the moment, I want us to focus on the cerebellum. Oh, sorry, even I just got it confused. The cerebrum, which is that big outermost layer, thicker region on the top that again looks like those like folded sausages. So the cerebral cortex, so we're getting more specific now, is the outermost layer of the cerebrum. So the cerebrum is all of the sausagey kind of looking bit at the top, and then the cerebral cortex is the outermost top layer of that. It's a folded structure, like I said before. The mountains are like the gyrus, so like the upper bits of the fold, and then like the valleys are known as like sulci, or they can also be referred to as like grooves and fissures. So we have the top bit, which is the cerebrum, and then we have the very top layer of that, which is the cerebral cortex, which is also kind of divided into two halves, which is our cerebral hemispheres, which you might be familiar as well. And they're also separated by the longitudinal fissure, which runs down the middle. So we have our left hemisphere and our right hemisphere as well. I'm not sure if that came up the right way on the camera or if I did that the right way, but we do have a left and a right hemisphere. So each hemisphere also has 4 lobes, which are mirrored on each hemisphere. So we're going to be looking at those in a second. There's also some other important parts of the brain that aren't on this diagram, such as like the basal ganglia, the primary motor cortex, and we do have the cerebellum on here. Those 3 you learn about, especially in relation to voluntary movement. And then the limbic system and prefrontal cortex you'll probably learn about in relation to emotion. So keep in mind there's a whole bunch of different parts of the brain that we need to be aware of. So as I mentioned before, there are those 4 different lobes, like that outermost layer of the cerebrum is the cerebral cortex. So we have the left and the right, and then that's broken up into these 4 different lobes. One thing that I think is really important is actually being able to label and remember where each are. So some of the tips that I have for how to remember that is, first of all, the frontal lobe. I find that fairly easy to remember because I know it's the frontal lobe. And then the other one that I find a little easy to remember is the occipital lobe. I remember that that's right up the back of the brain. How I remember that is because once you learn a little bit more about visual perception as well, that'll get a little bit easier because your eyes actually receive the sensory, like the stimuli from everything around you that you're seeing, and then they actually send that to the back of the brain for processing in the occipital lobe. So I find it fairly easy to remember frontal lobe's at the front and occipital lobe is at the back. But then sometimes it can be a little bit confusing as to how to remember where parietal and temporal lobes go. I kind of have a few little ways to remember it. One of the ways, it seems a little bit silly, but one of the ways that I like to remember it is I think about like a parent, like patting a child on the head. So I think like parietal lobe, like parental, like a parent patting their child on the head. Or you might think like temporal lobe is down a bit lower, so maybe it's near your ears because temporal is to do with like your sound and hearing. And then the last little trick that I have is I just think of like the letters PT, like a personal trainer. I think like P on the top and then T down below. So parietal lobe, temporal lobe. So that's kind of another little way that I remember it as well. Maybe one of those ways will make sense to you, maybe not. Whatever little way you can come up to remember those with will be really helpful to kind of, get it all straight in your head as to which one is where. So what do each of these areas actually do? So first up, the frontal lobe. That's like the bigger area. It's the one up the front. It's responsible for quite a lot of different things. It's responsible for like speech and language, abstract thought, movement, planning, judgment, problem solving, aspects of personality and emotion, and social skills. It's mostly like that higher order functions and like higher order thinking. But remember and keep in mind that the brain is very complicated. Quite a lot of the things that we try and like simplify, like memory occurs here or emotion is controlled by this. We've actually simplified quite a lot of it. So a lot of it happens with coordination and connection with a lot of different brain areas because the brain is a very complicated place and we're only kind of exploring the beginning of it here. Like I said before, occipital lobes are responsible for visual perception. How I remember that, it's like occipital, like I think eye, or like optometrist. It's kind of like those same kind of like word parts, occipital, like referring to the eye. Parietal lobes are responsible for spatial awareness and sense of touch. So for example, like pain, temperature. How I remember that is like spatial. The second letter is P, so parietal spatial. It's not the perfect little cue, but it does help me remember it. And then also like sense of touch, pain, like parietal pain. So that's another way to remember that it's responsible for touch. And then the last one is temporal lobes, which are responsible for auditory processing. So listening to sounds and words, recognizing them, comprehending speech and all of that. So I think temporal, like tempo, like music, sound, auditory. So that's how I kind of link those in my head as well. So here are some other key areas in the brain that are involved in language processing. So just to really simply overview kind of roughly what they do. First one we have here is Wernicke's area, which is in the left temporal lobe, and that's responsible for speech comprehension. So making sense of words, giving meaning to them and all of that. Whereas Broca's area, which is in the left frontal lobe, is more for speech production. So actually the mechanical decision to move your mouth and produce the words. And yeah, that mechanical production of actually producing speech. And then Geschwin's territory is a little bit more complicated. It's located in the parietal lobe, and I kind of think of it as like connecting the two areas because it kind of like processes all aspects of speech and brings it all together. So it brings together like the meaning, the sound, like the visual written shape of the words, and it kind of connects it and brings it all together in a way which is connected, which is kind of illustrated in this diagram here. Another interesting thing is when something actually goes wrong with one of these areas. For example, if something goes wrong with Broca's area, like it's referred to as Broca's aphasia, or this particular example we're looking at, we know that Broca's area is responsible for speech production, like that actual production of the words. So if someone has Broca's aphasia, their speech isn't fluid. Their speech isn't fluent. It's very like disjointed. They can't get their words out. They know what they're thinking. They know what they mean, but they actually can't get it out because there's like they're having trouble with the area of their brain that's responsible for actually producing the words. On the other hand, when we have Wernicke's area, which is responsible for like the comprehension and the meaning of speech, if somebody is experiencing Wernicke's aphasia, they're actually going to have impaired comprehension. So they're going to have no issues with like producing the speech. Like they could talk as clear and as quick as they would like with a normal rhythm of speech, but because there's that impaired comprehension, it actually doesn't carry like as much meaning. So I've heard that referred to as kind of like a word salad, because when they have Wernicke's aphasia, like their speech is unimpaired, like they can talk at a regular rhythm, but there's actually no meaning there. So the idea of like a word salad is they're just saying random words, but it actually doesn't make sense because their comprehension is quite impaired. So it's really interesting to think about when we know the normal anatomy and physiology of a certain area of the brain, we can actually start to kind of problem solve and like connect the knowledge and work out. So if something goes wrong with this area, if we know in a normal, healthy individual what that area does, then we can kind of start to logically work out what problems they're going to encounter if they do have an issue with that area. So next thing we're going to be looking at is neurotransmission. So I talked about the big picture of how we collect sensory information and like it's sent from our peripheral nervous system to our central nervous system, and then all of that processing and decision making, and then it sends back like a decision. But how does that actually happen? Well, that happens through the process of neurotransmission. And what you're actually asked to be able to do by the syllabus is communicate neurotransmission using a diagram. So you need to be able to understand the process, but also be able to construct and identify like a visual diagram of the process. So you need to be able to like label a neuron, discuss what's going on in the synapse, and all of those types of things. So this is an example of a diagram that we have here. You can see we have like the presynaptic neuron here, pre meaning like before, synaptic meaning synapse. So the neuron before the synapse, and then here we have the synapse, which is the gap between the two neurons. And then here we have the postsynaptic neuron, which is post after synaptic synapse, so the neuron after the synapse. And we can see this like zoomed into this little diagram here. But I am going to go to my next slide, which has a little bit more details about it. So again, these slides are available if you want to download them and have a look at this in a little bit more detail. So this is a diagram of a neuron that I've actually drawn and like labeled in quite a lot of detail. Probably one of the things I'm most proud of from my own senior psychology notes. But what I find really valuable about this diagram is it actually communicates like the broader big picture of like neurotransmission, which is like this electrochemical message with like a directive or information contained in it. How does that message actually pass from neuron to neuron so that we can have that communication within our nervous system? But then also, like the value of this having this little box here, which we had on our previous diagram as well, is we can kind of in essence do that like zoom in and show in detail what's actually happening. So it's also really important to make sure that you like know all of the terminology from a neuron so that you can like be able to label that if you're asked to. So for example, we've got like the nucleus, the dendrites, the cell body, we've got like the axon and the myelin sheath, which is that like fatty insulating like protective layer that like helps the messages like get sent, which goes down the axon and then to like the axon terminal, and then we have the synapse, the gap between the terminal and then the next dendrite, where the chemical message is passed along to the next neuron and that message continues. But just to briefly go into a little bit more depth, the first thing I want to draw your attention to is the process of synaptic transmission, which is a little bit different to neurotransmission. So neurotransmission is kind of the term for like that broader big picture. How is my message communicated from the first neuron to the second neuron, how it goes all the way along. But synaptic transmission is kind of that zoomed in process that I have highlighted in that box there. So what's actually happening in the synapse? How is that message actually getting from the end of the presynaptic neuron to the start of the postsynaptic neuron? So synaptic transmission, like we said, that process where the chemical messenger is a neurotransmitter. So a neurotransmitter is a chemical that like communicates that message, and that's where it crosses the synapse from one neuron, or it could also be referred to as a nerve cell. So if you hear neuron or nerve cell, we're thinking the same thing. And it's this process, this is how the nervous system communicates. So the steps in neurotransmission, in synaptic transmission. So what happens is these neurotransmitters, so these little chemical messengers, they're contained inside vesicles within the terminal buttons, or like you might hear it referred to as the axon terminal or the synaptic terminal of the presynaptic neuron. And what they do is they actually move towards the membrane. So they're moving towards like the outside of the neuron. And then the vesicles actually like break open into the synapse, and the neurotransmitter is actually released. And so once the neurotransmitter is released, you can see here we have our like presynaptic neuron, and we've got like the axon terminal. The vesicles are moving towards the membrane so that the neurotransmitter chemicals being released. And once it's released into the synapse, this like gap between the neurons, the neurotransmitters can then like bind with the receptors on the postsynaptic neuron, which initiates the continuation of the communication in the next cell. So that's how our message kind of continues. So this little chemical is like communicating what the message is. So once it binds here, that message can then continue in the next cell. So that's how we have this like ongoing communication. Like I said earlier, neurotransmission is kind of the broader picture, which involves like the electrical signal passing down like the axon of the neuron, and it also involves that synaptic transmission, where the neurotransmitter is released into the synapse, and then this neurotransmitter either excites or inhibits the neuron. So if it excites, it's like telling it to do something, or if it like inhibits, that's kind of like when it's like preventing, like no, don't do this. So it's kind of like that positive and negative effect. You might have learned about glutamate and GABA in relation to that. And then so if the neuron is sufficiently stimulated, which is when the threshold potential is reached, it'll send an electrical signal down the axon and the message will continue. So again, it's that whole process of how is this message actually traveling through. And you actually do need to be able to draw this, because like what I have here, the syllabus actually explicitly says you need to be able to communicate neurotransmission using a diagram. And using that little zoom in that I've shown you here is also a great way to communicate this knowledge, to show that you know what's happening in the bigger picture. You know that the direction of impulse is kind of always moving. From like the dendrites and like the cell body, like down to the axon terminal. I've also heard it referred to as like this is the head and this is the butt, so it always goes, or like the feet, so it always goes from like the head to the feet. So there's all of these other like little illustrations that you could use as well to help you really remember what all of the different parts are. So like what I was just talking about before, we actually have these neurotransmitters, which are these chemical messengers that are released within the nervous system to communicate between neurons. And here are 5 examples of neurotransmitters. Some of these are also hormones as well. And you actually need to be able to compare the physical and psychological functions or effects of each of these. So I'd recommend like doing some research, referring to your notes and all of that, and trying to come up with like a really solid, like, for example, like a table where you compare them, where you talk about their similarities and differences and what each of them do. So you need to be aware of like what effect they have on the body, like on the brain, and like a person's like psychological state, for example. Another thing that I'll quickly draw your attention to is the difference between neurotransmitters and hormones. So they're both chemical messengers, but the main difference is neurotransmitters are released within the nervous system, so they communicate within the nervous system, whereas hormones are released like outside of the nervous system. So some chemicals can exist as both neurotransmitters and hormones. So also being aware of where they're released and what that means for whether they're a neurotransmitter or a hormone. So the first one on our list is acetylcholine, which is a neurotransmitter that's found in like the brain and the spinal cord, and also in the peripheral nervous system. And it's involved in muscle contractions. So for example, like the lungs and the diaphragm to like help us breathe, as well as muscles involved in like digestion and cardiac muscles involved in like beating the heart. It's also involved in like learning and memory, as well as REM sleep. So it's important to be able to discuss some of those functions of acetylcholine. And we'll also shortly be looking at an example of what happens when something goes wrong with acetylcholine. So by knowing what acetylcholine does, it makes it much easier to discuss what happens when it goes wrong, which is in the case of Alzheimer's. Our second one is epinephrine. If you haven't heard of that term before, you might have heard of adrenaline. The important thing to remember is that epinephrine and adrenaline, one and the same, just different names. So epinephrine or adrenaline is both a hormone and a neurotransmitter, and it's involved in like the fear response. So like fight, flight, freeze, and it's regulated by the autonomic nervous system, and it involves like increasing the heart rate and blood pressure. So again, it's about like mobilizing the body to act. Like you're in an emergency situation, so you need to be able to deal with it. A good way to remember that is thinking about like, what does adrenaline actually do? So think about like, in movies or in your own life when you're like, oh my goodness, like I've got such an adrenaline rush. Like that's when you're in like a really dangerous or scary situation and your body is like super heightened, and your body actually physically reflects that. So that increased heart rate and blood pressure is so that you can get more oxygen and more glucose around the body, so that your body's like ready for action. And also like the increased respiratory rate, so you can get more oxygen to your muscles so that you're ready to like use that oxygen and respond to that dangerous situation. Epinephrine is released from the adrenal glands above the kidneys and also in the brain. And the important thing to know about this as well is that it occurs in intense bursts, which makes sense in light of what we probably know or have heard about adrenaline. Our next one is norepinephrine or noradrenaline, and is actually quite similar to adrenaline in a lot of ways. Also hormone and neurotransmitter, is also involved in mobilizing like both the brain and the body to act, so like the fight, flight, freeze response. Again, increasing heart rate, triggering the release of glucose into the bloodstream, increasing blood flow to the muscles, increasing alertness, arousal, regulating mood. But here's our main difference. And again, because we're looking at the similarities and differences between these hormones, it's important to be able to make that differentiation. The main difference is that noradrenaline or norepinephrine, it's more of a slow release chemical that's released continuously at low levels. So how I think about it is if you're in a super emergency, heightened situation, you're going to be getting a massive rush of adrenaline, but adrenaline or epinephrine, but that cannot be sustained for long periods of time. So the norepinephrine, noradrenaline, is kind of that slow release chemical, and it's actually continuously being released at low levels. So that's more of the level kind of hormone. It does similar things, but it's more released constantly at those low levels. Our fourth one is dopamine, which is again hormone and neurotransmitter, which means that it's present both inside and outside of the nervous system, and it's involved in our thoughts, feelings, pleasure, motivation and behavior, learning to associate a particular behavior with a reward, and also a little bit of it's associated with movement as well, which is also really important because we're going to be also having a look at what happens when something goes wrong with dopamine. Serotonin, our last one, hormone and neurotransmitter, again involved in regulating mood, especially happiness and well-being, as well as appetite and digestion, especially satiation, which is that feeling of feeling full, the sleep-wake cycle, memory, sexual functioning, the constriction of smooth muscles as well. So for example, the slow automatically constricting muscles in the walls of your blood vessels and organs, for example. So yeah, those are just some of the functions of each of those. Again, I'd encourage you to make a table of the physical and psychological effects of each of them, and then also the similarities and differences and their effect on, again, the brain and body. So like I said before, we're going to be having a look now at interference in neurotransmitter function. So we've briefly reviewed how do neurotransmitters work, what do they do, they send those chemical messages, but what happens when something goes wrong? Because like I mentioned before, when we know what the hormones do, it's really easy to know a lot more and be able to problem solve. Well, when something goes wrong with this hormone, what do I think my expected symptoms are going to be? So the two examples that we're going to be looking at are Parkinson's and Alzheimer's disease, which are neurodegenerative conditions, which are diseases of the brain that progressively get worse with time. So you have a progressive loss of both structure and functions of neurons in the nervous system. So in these conditions, the person's nervous system is actually quite deteriorating. So they're losing a lot of structure and function, which can be a very confronting thing to see, especially if you've had a family member suffering from these conditions. So the first one we'll be discussing is Parkinson's disease, which you may or may not be familiar with. The symptoms include loss of motor control, which is most noticeable by the fact they might have a little tremor. That's starting to indicate that they're losing that a bit of their motor control. It can also result in a depressed mood, decreased cognitive function. So for example, impaired memory and reasoning and those thinking, as well as difficulty sleeping. It can cause pain as well as reduced facial expression. And now we're going to switch gears a little bit and think about what's actually causing this. Obviously, these are quite complicated conditions and there's a lot of factors that go into them, but specifically using our knowledge of neurotransmission, we're going to be thinking what's going wrong in neurotransmission that might be contributing to this condition. So in the case of Parkinson's, the mechanism for motor loss is actually a loss of cells responsible for producing dopamine in the substantia nigra. The substantia nigra is part of the basal ganglia, And we've just been learning about dopamine, which is a really valuable connection. So dopamine helps control messages as they pass between the substantia nigra and striatum. And the striatum is responsible for balance and control of movement. So we do see the symptoms of the patient not being able to adequately control their movement. So for example, like the tremor might become noticeable, and as the disease progresses, like a greater loss of that ability to control movement. And because dopamine, from our discussion on the previous slide, we also know that dopamine is implicated in movement. Again, those symptoms make sense. Treatment options, because the syllabus also does refer to treatment options. They include dopaminergic medications. So for example, a medication that gets converted to dopamine in the brain, like L-Dopa, or medications that like mimic the action of dopamine by acting on dopamine receptors, which links in really well with neurotransmission. Because if our problem is being caused by like issues with our dopamine, then medications that mimic dopamine or try and create more dopamine are going to hopefully help reduce the impact of this issue and reduce like the severity of the symptoms. Or another thing we can do is administer a medication that like prevents the breakdown of available dopamine in the synapses. So a little bit of background knowledge behind that. When a neurotransmitter is actually released into the synapse, it acts on the receptors of the following neuron, but we actually don't want that neurotransmitter to stay there forever, or the same message is going to be sent and sent and sent. That neurotransmitter actually needs to be cleared away. And how that's done is by either releasing like further chemicals that like break down that neurotransmitter to get rid of it, to get ready for the next message, or reuptake of the neurotransmitter. So for example, some chemicals like serotonin are released into the synapse, and then they're taken back up into the cell. You might have heard of like SSRIs, which I can't remember exactly, but I think it's like selective serotonin reuptake inhibitor. So the idea of that is we're saying, no, don't bring the serotonin back into the cell. Like I actually want to leave it in the synapse so it can continue to have an effect. So again, the idea of if we give someone a medication that prevents the breakdown of available dopamine in the synapse, we can hopefully counteract that issue of inadequate dopamine. So that's kind of the thought process there. Other small things that you could discuss are just like exercise, diet adjustments, but also deep brain stimulation, which is a very like invasive procedure where electrodes are placed in the brain, or forced ultrasound therapy, where like ultrasound beams like target the thalamus. So there's a few different like treatment options, but again, Parkinson's is a very serious and complicated condition. And it's not quite as straightforward as we make it out to be here, but they still are options and things from a psychological perspective that we can consider. The next thing that the condition we'll be looking at is Alzheimer's disease, which again, you may or may not be familiar with. The symptoms include like memory loss. It also interferes with a person's ability to form new memories, which is like anterograde memory loss, and also the loss of old memories, which is that retrograde memory loss. It also results in like confusion, impaired attention, disordered thinking, depressed mood, and some other symptoms similar to these. And again, coming back to the idea of what's actually like the pathophysiology of this, what's actually causing this disease. Well, part of it is actually caused by like protein buildups in the synapses. So again, the synapse being like the gap between the neurons. So protein buildups in the synapse, known as beta amyloid plaques, cause low levels of acetylcholine and other neurotransmitters. So we're linking in here what we know about acetylcholine. So these plaques increase the activity of the enzyme acetylcholine esterase. An important thing to know about that is when we're talking about enzymes, you can kind of identify an enzyme by its like suffix ASE. So acetylcholine esterase is an enzyme that is responsible for breaking down acetylcholine. So coming back to what we know about Alzheimer's, like the protein buildups in the synapses, these beta amyloid plaques, they cause low levels of acetylcholine and other neurotransmitters. So part of our issue here with Alzheimer's is that we have low levels of acetylcholine. We also recently just learned that one of the things acetylcholine is implicated in is that like memory and learning. So these low levels of acetylcholine are going to be causing issues with memory and learning, which are the symptoms that we are seeing in Alzheimer's. And again, I spoke about earlier about how neurotransmitters are released into the synapse. Sometimes they're like broken down or like reuptake occurs. Well, in the case of like acetylcholinesterase, that's an enzyme that's responsible for breaking down the acetylcholine to stop that message being sent. But again, these plaques increase the activity of that enzyme. So these plaques increase the activity of that enzyme, which breaks down acetylcholine. So you can see how these plaques are causing that lack of acetylcholine. So these plaques, again, all leading to the idea that we have less acetylcholine because it's being broken down more. So this lack of acetylcholine is responsible for it's responsible for learning and memory, which is how we get these symptoms. The other thing mentioned here on this slide is neurofibrillary tangles, which are proteins inside the neuron, whereas our beta amyloid plaques were in the synapse. Our neurofibrillary tangles are proteins inside the neurons, and they actually cause cell death. They start in the hippocampus, which is responsible for memory, and then they do progress to other areas of the brain, which is again going to be causing more issues. Hippocampus is also implicated in memory, so we're going to see further of those issues involved in memory. Treatment options, again, medication that inhibits cholinesterase, or like that acetylcholinesterase, which is again enzymes that break down acetylcholine. So if we can stop the chemicals that are trying to get rid of the chemical that we want to keep, then that's an awesome thing. So we're going to be able to keep more of the acetylcholine, which is what we want. It's important for memory and learning and healthy functioning. Or also medication that regulates the activity of glutamate, which is also involved in information processing and learning. So it's interesting how we can kind of link our knowledge about neurotransmitters to what happens when it goes wrong. What do we see and how could we possibly go about fixing it? So here we actually have a sample question. So it says, Finn was standing near a campfire with his friends when he noticed the fire becoming hotter against the skin of his legs. To avoid getting burnt by the growing flames, sorry, the words are just a little bit small here, he took a step away from the campfire. The human nervous system has two major divisions, which again, we should be thinking of like the central nervous system and the peripheral nervous system. So identify the subdivision of one of these major divisions. So now we're thinking about a subdivision of one of these divisions, which in my mind, I'm coming back to the fact that the peripheral nervous system has like the most subdivisions. So identify one of these major subdivisions that activates Finn's response, and outline how the subdivision is involved in his response. So this is a sample question from an old psychology exam. So if you would like to take note of this or pause the video, you will be able to have a try at this. But now we are going to have a look at a sample solution. So this is from the examiner's report, where they've actually broken down how people approach the question, what were acceptable answers and what were also like common mistakes. So we're going to have a look at that here. So there's lots of information on this slide here. So how they've broken down the question, they've said there were 3 valid interpretations of the questions. Full marks could be achieved by providing an accurate identification and a correct and coherent outline of the involvement of any one of the following in activating the response. So they've said that it's okay for people to talk about the role of the somatic nervous system, the brain, and the autonomic nervous system. So again, when it's talked about how there's those 2 major divisions in the central nervous system, first one being like central nervous system, brain and spinal cord. So they said you could talk about the brain's role in his response to that. Or when we're thinking about divisions of the peripheral nervous system, it would be okay to talk about autonomic or somatic. But the main thing, again, you can read this information that I have on the slide here. The main answer that they were looking for is, I believe, the somatic nervous system, because it wasn't a super emergency situation, and he was consciously and voluntarily choosing to step away from the fire. So in this scenario here, the somatic nervous system does make sense because he detected the stimulus on his leg and then he voluntarily chose to step away from the campfire, which is again part of like the somatic nervous system, that interaction with the external environment, control of muscles, and that voluntary conscious decision. And so he chose to make that response. And there's also some information on this slide about if people justified their answer for the brain or autonomic nervous system, then those answers were also accepted as well. So now we're moving into topic 2, visual perception. So visual perception is all about, or in the name, visual perception, how we perceive visual stimuli. And there's two main like overviews, like two main overarching parts of this. Sensation is like the first 3 steps. So sensation involves reception, transduction, and transmission. And then our second 3 are part of the step that we call perception, which is that selection, organization, and interpretation. So to give a bit of background knowledge for this, like when we see something, when we see anything around us, the image on our retina is actually upside down, back to front, it's blurry and it's patchy. So these 6 stages cover from like the time that our eyes are actually collecting the sensory information to when our brain actually interprets what we see. So sensation, our first one, sensation being kind of that overarching first 3, when we're like sensation, we're receiving the sensory information, and then perception is kind of the second 3, when we're perceiving it, we're making sense of it. So sensation, the first 3 parts. The first one is reception. So light energy is collected by our eyes. It enters through the cornea, it travels through the pupil, and is focused on the retina at the back of our eyes. So this light energy, and light energy is like the visual part of the electromagnetic spectrum, and it must be within like the visible light spectrum, which is around 360 to 760 nanometers, and that's for it to be perceived by the average human. So reception is all about light energy being collected by the eyes. That's the basic overview of it. Our second step, transduction, is where light energy is received by the receptive fields at the back of the eye in the retina. So the receptive fields are groups of photoreceptors, which again are cells on the back of the retina that are sensitive to and they detect light. We have 2 types of photodetectors that are working. We have rods and we have cones. So rods are particularly sensitive to like black and white, and we rely on them more in dim light. They're very light sensitive and they're responsible for like a lot of our like peripheral vision, and they're concentrated more at the edge of the retinas, and they have like very low visual acuity, so which is kind of like low detail and sharpness, which is contrasted with our cones, our second type of photoreceptor cells, which provide clear vision. Like they're all about color vision. They work really well in the bright light, and they're concentrated in the middle of the retina. They're responsible for visual details and require high levels of light to enable a response. So in this step of transduction, the photoreceptors, which are our rods and our cones, again, rods, black and white, cones, color vision. That's another good way to think about it. Cones, color, whereas the rods are the black and white vision. What they actually do is they convert this light energy, this incoming like stimulus information, to electrochemical nerve impulses that can be passed through the optic nerve to the brain in our third step, which is transmission. So it's about taking this light energy and putting it into an electrochemical message that we can actually send to the brain. And transmission, the third step, is when this electrochemical message is passed along the optic nerve to the visual cortex, so the brain can actually start to make sense of what we see. Because without our brain, our eyes would be useless, and the visual cortex is in the occipital lobes of the brain. again, up the back of the brain. So that's another way to help remember what the occipital lobe is responsible for, visual perception. So those first 3 steps are sensation, so when we're receiving that sensory information from the environment, like the light energy entering our eyes, being focused on the retina up the back, being converted to that electrochemical message, and then that message is sent along the optic nerve to the brain. And then this is where we enter perception. The next 3 steps, when we start to make sense of what we see. Step number 4 of 6, but like the first part of perception, is called selection. So this is getting into the idea that we actually can't pay attention to like the millions of stimuli that we receive at the same time. So our brain actually picks what we pay attention to, and the optic nerve actually has a role in it as well. So think like selective attention. Unconsciously, we're choosing what to pay attention to. So this is actually done by feature detectors in the optic nerve and visual cortex, which break up the images into their features. So for example, like line length, to help us decide what are we actually going to pay attention to. A really practical way to think about it is we're actually never paying attention to everything that we're looking at once. So for example, currently I'm focused on like my laptop screen and my camera as I'm like delivering this presentation, whereas behind my laptop I actually have my whole bookcase, but I'm not actually actively paying attention to any of the titles and words because my brain actually can't actively process all of this information all at once. So my feature detectors are helping me like decide and undertake selection. What should I actually be processing and taking notice of? Which brings us to organization, our 5th step, where the visual cortex recognizes the information and it organizes it into a meaningful whole so that we can actually make sense of it using like visual perception principles. Some of these are like our perceptual constancies, Gestalt principles, depth cues, which we'll look at later. So these visual perception principles help us guide the reassembly of the image. So we're kind of putting the image together in our mind, which brings us to interpretation, the 6th step, which is where we actually make sense of it and give it meaning. So we can the image is kind of put together in organization, like it's being organized, but it's not until our 6th step, interpretation, where it's actually kind of given meaning to, like we make sense of it. And how it does this is it's simultaneously sent to the temporal lobe, to identify the object by comparing it to information we already have in memory. So for example, if I see the face of somebody I'm familiar with, that information is entering my eyes, being sent to my brain, and then my occipital lobe is sending it to my temporal lobe and comparing it to what I already have in memory. Like, oh, that's Michelle, because I'm comparing it to information I already have in memory, so I can recognize it. Even something as simple as an apple, I can recognize an apple because I'm comparing it to pre-existing memory. And simultaneously, that information is also sent to the parietal lobe to judge where the object is in relation to our visual field and ourself. So again, parietal lobe, we're thinking like spatial awareness kind of thing. So that makes sense because we send the information to the parietal lobe to judge where it is, like in distance wise, like is it close, is it far? And again, we use depth cues and all sorts of things to help us with that. And then images are also given meaning based off our perceptual set, which we'll be having a look at shortly. So that's kind of things like our past experiences, like our motivation, our values, and our context. So we're kind of getting into the idea that there are so many different things that influence how we see what we see. So there's this like biological process of we're actually getting the image that we see into our brain so that we can actually process it. But then there's also all of these other things that are influencing what we see, which we're going to be having a look at now. We're going to be looking at like some of the biological, psychological, and like social and cultural impacts or influences on visual perception, which is getting into the idea that there are quite a lot of things that influence our visual perception. So things that influence how we see what we see. For example, even just like a really random example, when most people look at a dog, like that might bring them like really warm and happy feelings because they might think it's a really cute dog. But somebody that has the past experience of being attacked by a dog might go like, oh my gosh, and enter like absolute panic mode, which is a completely appropriate response based off their past experience. But you can kind of see how our visual perception may differ based off a whole lot of different things. This is just a diagram of like the pathway of light through the eye. So light enters through the cornea, travels through the pupil and like the lens, and then it's focused on the retina at the back of the eye, and then is sent along the optic nerve to the brain. So again, if you do some research or review your textbook, I'm sure you'll be able to find a much more detailed diagram that might help you with like the anatomy of the eye. Again, we have those two parts, and our second kind of three are like that selection, organization, and interpretation, which is like our perception. And again, that's all about making sense of what we see. So how can we go from seeing a bunch of mismatched random pieces to how can we actually make sense of what we see? Awesome. So first kind of influence that we're looking at with influences on visual perception is biological influences. So biological influences, the first thing is actually the physiological makeup of the eye. So think about the physical structure and function of your eye. If your eye is physically damaged, that is a biological structural thing that's going to affect how you see things. But another thing we talk about there is like flexibility and shape. So again, how your eye is physically structured and made up is actually going to influence how you see what you see. So that is our first biological influence on visual perception. The second one that we actually look at quite a lot is aging, and how we kind of think about that is like a big influence of that is actually like a loss of elasticity in the eye. So as we age, our eyes actually become less flexible. And so because of that, it actually is again influencing like what we're able to see, which is really, really interesting. So to go into that a little bit more, as we age, our eye loses elasticity and flexibility, like I said before, and that's kind of like because our lens is hardening with age. So we can no longer like bend it or like alter it as easily. So we can't actually like bend light as accurately to focus it on the retina like we could before. So because again, as people age, their eye is like losing that elasticity and flexibility. The lens is getting harder. Normally, depending on whether we're focusing like up close or far away, like our lens actually like bulges and stretches to like accommodate that. But if it's like hardening, it can't actually do that anymore. So it can't actually focus the light on the retina as it should or could in like a healthy, like a younger individual potentially. So that like becomes an issue. So it might be focusing the light like too soon or like too far back. So it can cause issues on like people seeing things like near or far away, depending on like how the lens is changing. And this is called like presbyopia. And there are also other things such as like floaters or cataracts, age-related macular degeneration and glaucoma, all of which like reduce a person's vision and they all increase with likelihood as we age. Our third biological influence on visual perception is actually our genetics. So here we're thinking about inherited visual disorders that are passed on from parent to offspring through their genes. So congenital visual disorders are present from birth. So for example, color vision deficiency is a big example that we look at here. And depending on whether it's like monochromacy or trichromacy, for example, is going to depend on the extent to which they're colorblind. Some people are like red-green colorblind, but there also are like rare cases where people are like completely can only see in like grayscale because they have no color vision, which is which would be quite a challenging thing. So again, to link that back with our previous knowledge, we talked about how the photoreceptors, the cells on the back of our retinas, we had our rods and our cones. Our cones were responsible for like that high quality color vision, and our rods were responsible for the black and white vision. So what happens in color blindness is people might be missing some of the cones, so they actually have the inability to perceive that color. So again, making links between related content is really helpful when you're trying to deepen your understanding of the content when studying and like making those interrelated connections, which is something that we'll talk about in a lot more depth when we get to memory as well. Next, we're going to be able to look at Gestalt or like Gestalt principles, which these are what we referred to a little bit earlier when we were talking about we have these different principles which actually guide how we perceive the things that we see. So these are classed under psychological influences on visual perception. And one of the subcategories of that is these Gestalt principles. So what are Gestalt principles? They are based on the idea that the whole is greater than the sum of its parts. So this means that our brain attempts to perceive visual information as a meaningful whole. So instead of seeing smaller isolated parts of information, our brain attempts to create like a whole picture. Like, for example, this smiley face in the bottom right-hand corner. I don't just see all of these individual little dots. My brain actually sees it as a meaningful whole because of these different principles. Because our brain, in essence, is trying to fill in gaps and say, I don't want to just see all the random parts. I'm going to fill in the gaps, I'm going to make a guess, and I'm going to see what I think I'm meant to be seeing. So the first Gestalt principle is figure-ground organization, which occurs when we focus on an object visually. So it's called figure-ground organization. So the first part, the figure, is the object that we're focusing on, and it's separated from the ground, or like the background or surrounding objects, by a contour line owned by the figure. So for example, Rubens' vase in the top right-hand corner, or the Apple logo. So it's the figure, what we're focusing on, is separated from the background by that contour line. An interesting side note, camouflage is actually a special type of figure-ground organization when the contour is deliberately, like that line is deliberately blended in with the background. And the goal of camouflage is that the figure isn't meant to be perceived. The next Gestalt principle is closure, which is when we perceive an object as being whole, despite it actually being incomplete. So again, this closure is a massive instance of our brain filling in the gaps. So for example, in this image here, you might see a triangle. The interesting thing is though, is that there is actually not a triangle there. There is no white triangle, but because of how this image has been constructed, we think there is, because closure has been applied. Our brain has filled in the gaps and assumed that there is a white triangle there. The next one is similarity, which occurs when individuals perceive an object based on the parts that are similar. So for example, it might have elements that are similar in size, shape, or color, and so we perceive it a certain way due to that similarity. So for example, here in this image, I'm going to perceive like these diamonds. I kind of perceive this section as a meaningful whole, and then I perceive like the circles together because I'm perceiving it in a certain way due to the similarity of the individual objects. Or for example, this one here, these objects are all similar in color, so I'm perceiving them kind of more as like a meaningful whole. Again, it's just because of the similarity. I'm perceiving it a certain way because of the similarity. Or the last one, or another good example of similarity is here. I'm perceiving this as lines of dots or columns of dots instead of rows because of the similarity of like the color and structure. The last one is proximity, which is when we perceive something a certain way due to proximity. So for example, I'm perceiving these singular dots as like a continual smiley face line because of their proximity to each other. Their proximity is influencing how I'm perceiving them. Another psychological influence on visual perception are visual constancies, which are kind of like those predispositions that exist in our mind that actually enable us to maintain a stable perception of a stimulus, although the image size on the retina may change. For example, if someone is walking towards you, it might look like they're getting bigger, but you actually know that they're not. Their size is staying the same. We're just interpreting it as getting closer. So our brain is using that cue of their changing size to tell you, well, oh, they must be getting closer, to help us make sense of what we're seeing. For example, you probably just interpreted that my hand stayed the exact same size, it just got closer to my camera, because you have that visual constancy, that size constancy of, well, I know that her hand is staying the same size, it just must be getting closer. So you actually use that as a depth cue. How interesting. So the other interesting thing is, like, the more familiar we are with the stimulus, the more likely we are to maintain a perceptual constancies. And visual constancies are usually learnt in childhood. And we need to be familiar with the thing to know what it should look like, so that we can have that constant, stable perception of it. So for example, most people are very familiar with roughly how big a hand is. So you can maintain that constancy. But if, for example, I saw an image of something that, like, I didn't recognize and I'd never seen it before, and if there was nothing else for me to compare it to and trying to like figure out the size of, like, I actually wouldn't be able to like know what size it should be. So I wouldn't be able to maintain that perceptual constancy. So again, that prior knowledge, it's different for every person, like different for people in different cultures and different families, that might vary as well. So to quickly reflect on these a bit more specifically, like we just talked about a few examples of, size constancy is that like ability to maintain a constant perception of an object. So it might seem to get bigger, but we just know it's actually getting closer. We don't change our perception of the object. We just use this changing size of the image to judge distance, in this case, for example. Another one is shape constancy, which you might not have thought of as much before, but that's when we perceive an object to maintain its known shape, despite like the changing perspective from which it's observed. So for example, opening and closing a door. We actually maintain the shape constancy. I know that this door is still a rectangle. But the interesting thing is, when you open and close the door, it actually changes back and forth. Like the 2D image on your retina, as the door is like pushed away from you, it actually appears to become a trapezium. But we know it's still a rectangle. We just know that the door is being opened. I'd encourage you to try that later. Open and close the door and challenge your brain to think, is this a trapezium or is this a rectangle? Interesting trick for your brain. The other thing is brightness. The shade or brightness of something doesn't change even if the level of light it's exposed to changes. A really good example of this is your phone screen. If you are in like the blinding sun, you can like barely make out what's on your phone screen. But if you're in a dark room, like the light's probably going to blind you from your phone screen. But even though it seems to change, we have that visual constancy of, I know the brightness isn't actually changing, it's just my perception of it because of these external influences. It's very interesting. Another example, what do you see here? My guess is probably people. And what do you see here? My guess is probably animals. The interesting thing is, those last images were almost the exact same. This is kind of known as like the rat man illusion, depending on where or ambiguous figure, depending on what you focus on, and just depending on like the way I was like priming your brain by showing you either people beforehand or animals beforehand, can actually change your perception of the image, which is so interesting. So visual perception, what we're looking at now is perceptual set. So this is another part of psychological influences on visual perception. So this is referring to how your past experience, context, motivation, and emotional state can be a psychological influence on your visual perception. So what is a perceptual set? It's a predisposition to perceive stimuli in a certain way according to our preconceptions. It's created by past experiences and it influences perception. So for example, what I just showed you before with Bujelski and Alampe's rat man experiment. By showing you people or animals beforehand, I was trying to predispose you to perceive it in a certain way according to preconceptions. So first one, past experiences. Previous exposure to a visual stimulus can affect perception, especially if it holds like a significant personal meaning. So again, we're getting into the idea that the same stimulus can be interpreted differently by different people because of past experiences. For example, if somebody saw like a white fluffy bunny, most people would think like, oh, like that's so cute. But for me personally, I actually had a rather scary encounter with a very scary bunny when I was a lot younger. So I kind of think bunnies are a little bit less cute. So that's the idea that based off our past experiences, again with my dog example earlier, people can perceive things differently. Also, context, the environment in which you observe it, can affect our expectations. So again, linking back to the rat man ambiguous figure I showed you earlier, the context of seeing it in people made you want to see it as a person. The context of seeing it amongst animals made you want to see it as an animal. Next, we have motivation, which is the idea that often we see what we want to see. So if on a long road trip, if you're really like desperately low on fuel and you see a sign that says food ahead, you might accidentally interpret it as fuel ahead because you see what you want to see. The last thing is emotion. So we can interpret things, our interpretation can be also influenced by our emotion. So for example, we could interpret somebody's face as like laughing or crying depending on our own mood. So have you ever looked at someone and been like, oh, I actually don't know if they're laughing or crying? This is the idea that depending on our emotional state, that's going to influence how we perceive them. And also this interesting image here, this is like a bunny, like a rabbit duck thing. So depending on where you focus, you might be able to see like a rabbit or a duck. Interesting. Another example of how we can see things differently. Awesome. Visual perception. Now we're going to be looking at more ways that we like perceive things. With the first one being the different monocular depth cues. So how do we actually perceive depth? We have monocular depth cues and binocular depth cues. So what is depth perception? Depth perception is the ability to accurately judge 3D space and distance. And we do this using cues in the environment. So the interesting thing is that our retinas can actually only see in 2D. I'm surrounded by a 3D world, but the image of what I see on the back of my retinas is only 2D. It's a 2D picture that we're actually seeing, which is why these depth cues are, again, they're so important for perception. And there are two types. Binocular depth cues, which is where we use both eyes together to gauge like distance and space, or monocular depth cues, where we can either use one eye independently or like it can be using both together. Because if, for example, you only have one eye open, you can't use binocular depth cues, but you still need to be able to have a sense of like distance and space. So the first monocular depth cue is accommodation, which involves the eye's lens changing shape so it can focus the light rays on the retina at the back of the eye. So we have these little muscles called ciliary muscles, and they control the movement of the lens. So the brain receives this information of what the ciliary muscles are doing, and it actually like interprets the tension in the ciliary muscles to determine distance. So depending on how like our eye is moving and the accommodation of our lens and ciliary muscles, our brain can actually use that information to kind of judge how far away something might be. The next main one is pictorial depth cues, which are used by artists to create a 3D interpretation of something that exists on a 2D surface. So these pictorial depth cues are how we kind of interpret that something's meant to like look 3D, even though it's technically a 2D picture. So the first one is linear perspective, which is the mild convergence of parallel lines as they extend towards like the distance. So what's interesting here, for example, like the road or the footpath, this line is kind of slightly moving in, and if we could see it, so would the edge of this road as well. Same with these footpaths. We know that these lines are like parallel, like the road and the footpath is running parallel. But the interesting thing is, if you look really closely, you can actually see that the lines are appearing to mildly converge and get closer together as it recedes into the distance. And that actually like creates the effect of it looks like it's receding into the distance. Even though, again, this picture I'm showing you, it's a 2D picture. These cues and everything is just what's helping your brain interpret it as 3D. And the linear perspective, like the lines coming together, is just one way that happens. The next one is interposition, which is just a fancy word for overlap. And that's when partially obscured objects are perceived to be further away, and the unobscured object is seen to be in the foreground. For example, the car is in the foreground and the trees are in the background. The trees are partially obscured. So I'm kind of thinking in my head, like, okay, well, if the car is in front of the trees, then the car must be closer than the trees. That's how I'm drawing that 3D conclusion. And that's really helpful for knowing which is closer, but it doesn't actually help me judge actual distance. Our next one is texture gradient, which is the idea that as the surface recedes into the distance, the texture or the detail decreases. So right up the back of the picture, there's a lot less texture I can see in the leaves, whereas the trees that are closer to me, I can see more leaves, I can see a lot more detail. So I'm interpreting it to be closer because I can see more detail. So I'm using that texture gradient. Next one is relative size. So based on our tendency to perceive the object producing the largest retinal image as being closer. So it's getting into that idea. So again, that first car is producing a bigger size on my retinal image than the last car. So I'm going, well, okay, size constancy, I'm referring to that knowledge. I know that those two cars are like a similar size. So the fact that that back car is smaller, it's telling me that it's further away. I know it's not actually smaller, I just know it's further away. So I'm using the knowledge of the real size of that object to make a judgment about, and make a comparison. So it's telling me which one's closer. And the last one is height in the visual field, which shows depth by betraying objects as farther away as being closer to the horizon, which you can see here, because our horizon is kind of like way back here. And this is closest to the horizon and it's furthest away. This is like a little bit lower in the visual field. So I'm perceiving that as like being closer, which again, it's really interesting how all of this knowledge kind of like is coming together to help me perceive this completely 2D picture, but to perceive it as 3D using all of that information. And that brings us to our binocular depth cues. So that's when we're thinking about like how our two eyes are working together to kind of like make sense of the world. So, and our two are retinal disparity and convergence. So the first one is retinal disparity, which occurs because our eyes are actually set 6 to 7 centimeters apart. My left eye is here and my right eye is here. They're actually 6 to 7 centimeters apart, and that's known as our pupillary distance. So the fact that our eyes, they're not in the same spot, they're different. So when an object is within 20 meters, so like close-ish, each eye perceives the image slightly differently, which is so interesting. But the brain fuses the two images together. So what we can learn from that is the more different the object is, the closer it is. If that doesn't make sense, that's okay. I'm going to try to illustrate that. So pick something like in whatever room you're sitting in, like pick something and preferably like a little bit far away and cover it with your thumb. Well, first of all, close one eye. So you should have one eye closed and you should be covering something with your thumb. And then what I want you to do is switch eyes. Open the eye that was closed and close the eye that was open. You'll notice the thing you were covering just moved. Well, it didn't actually move, but that's illustrating that our eyes actually see different things. So if you're ever looking through like a little like telescope or something, for example, if you swap eyes, what you see is going to be different. So that's showing that our eyes are seeing slightly different things. But when something's really far away, it doesn't like have a big of an impact. But when things are really close, it makes a big difference. So the more different the two images our eyes see, the closer it is. And the second one is convergence. So as an object gets closer, our eyes turn inward to keep it focused in the center. So for example, I've got my pointer finger up here. As I'm moving it closer, I can feel my eyes turning in because my eyes have to turn in to keep focusing on it in the center. And the brain actually reads the amount of like turning and angling in the eye muscles, and it uses this to make judgments about how close or far away the object is. For example, I can feel my eyes are really turned in when I'm trying to look right at my nose. But when I'm looking at something far away, my eyes don't need to turn as much because it's further away. So that's like another way that we gauge depth perception. So, social and cultural influences on visual perception. There's case studies about this which are really, really interesting. So again, visual perception is also influenced by social and cultural understanding. So we're just going to have a quick look at one example here. So, is the hunter trying to spear the tiny elephant or the gazelle? Most of you would say the gazelle, because you've used the knowledge of height in the visual field and relative size to judge, well, actually, obviously, I know that an elephant's a lot bigger than that, so it must be like further away. And because the gazelle's like bigger, it must be up closer. So obviously, our guy's trying to spear the gazelle. But it's so interesting. The researchers that did this study actually discovered that some African tribes don't see those pictorial depth cues of relative size and height in the visual field to estimate depth and distance. Our individual and cultural experiences influence what we see. So for example, like maybe it was because like those people like weren't familiar with interpreting 2D images or like weren't familiar with seeing stuff printed on paper. There's a whole bunch of like different strengths and limitations you can look at in these case studies, which I definitely encourage you to do a lot of research on. But so interesting that people in this like African tribe like assumed he was trying to spear a tiny elephant because they weren't used to using those same depth cues. So that shows you that that's not biological. It's not universal for all people. It's actually social, it's cultural, it's different between people groups, and it's influenced by our upbringing and what we're used to perceiving. The studies related to this are done by Hudson in 1960, Derogowski in 1972, and then the third study is Derogowski, Mulger, and Mulger in 1972. There's a lot of overlap between these studies, but a lot of them looked at quite similar things of like how the different groups of people actually interpret the same image differently, and there's a lot of really helpful information in those, so definitely do some research on those as well. Awesome. Visual perception. Our next kind of subtopic is actually the fallibility of visual perceptions, which is getting into the idea that our brain can be tricked really easily. So, question, which line is longer? The temptation might be to say line B, but they're actually the exact same. So visual illusions, which is what we're looking at here, visual illusions occur when perception differs from objective reality. So it looks different to what it really is. It's meant to be trying to trick our brain. There's kind of multiple interpretations and guesses and theories for why this happened. For the Malaya illusion, the carpentered room, like the carpentered world, I think it's like the room hypothesis is kind of like one theory for it. Another theory is like our brain fills in this gap and like interprets it as longer. So there's like different speculations for why our brain can be so easily tricked. But the bottom line kind of is that like our brain can be tricked very easily. Visual perception is fallible. Like it's actually not perfect. Another fun little activity, if you do want to try sometime, is if you actually take a piece of paper and if you roll it up, if you put your hand out like this and put the paper next to it and close one eye, and if you move it around enough and get it in the right spot, you can actually see through a hole in your hand, which is quite interesting. So try that with like a piece of paper. It might need to be a bit shorter than that one. But if you try that, you can actually see through a hole in your hand because your brain, your eyes are seeing different things, it's meshing them together, and it makes it look like you're looking through a hole in your hand. It's really interesting. So again, coming back to, you can trick your brain. Our brain doesn't always see what's actually happening. These two lines are the exact same length, which is really interesting. The next one, this is another visual illusion. It's the Ames room. So this is an illusion that causes people to perceive like people or objects of similar sizes as vastly different in size. So what we can see here is we grow up very, very used to, rectangle or square rooms that are bound by right angles. But what they did in the Ames room was they actually brought this corner up to make more of like a trapezium, like it's a trapezoidal room. So this corner is actually a lot closer. And then they got people to look into this room with one eye. So they could only use their monocular depth cues, and binocular depth cues are stronger. So it was like trying to trick them. And so the interesting interpretation was, well, what are people going to see? Like, can we trick their brain? And the answer was like definitely yes. So this demonstrated that we actually maintain shape constancy. In this case, our brain said, oh, the room is square, at the expense of size constancy. So what people see here when they look through that little hole into the Ames room is they go, oh my gosh, that person is giant. Oh, that person is tiny. Whereas they're actually about the same size. This one's just closer. But because we're so used to rooms being like rectangular and square, we're just interpreting that the closer person must be bigger because we are so used to rooms that are this shape. So again, it's like tricking our minds. So we perceive the room to be bound by right angles because of our lifetime of experience with rectangular rooms. So it's tricking us. Interesting thing, I don't believe like people potentially from different cultures who live at different places in the world that were used to like round mud hut rooms. I don't think they were actually as tricked by this because they weren't so used to like right angled rooms. So is that potentially another cultural influence on visual perception? That's again so interesting. So many different things that can trick us from seeing what we think we're seeing and like change how we think we're meant to see things. Ponzo illusion occurs when people perceive two lines of the same length as different. Again, two lines, same length. It uses linear perspective and height in the visual field to make it seem like the upper line is further away. And because the images are casting the same size image on the retina, we perceive the more distant line to be longer. So again, bringing in all of those different things. Again, we can actually have impossible figures. So have a look at this and try not to like boggle your brain too much. So what impossible figures do is they exploit perceptual cues so that we're actually faced with an image that should not be possible. So 2D figures which are visually interpreted as representing 3D objects. Maybe that's because we rely on visual cues so strongly. I don't know, potentially. We're still not sure why we see them this way. And then the other type of figures we look at are ambiguous figures, which is like what I showed you earlier with like the rat man. So they can be perceived in different ways. We have like a multi-stable perception of them. So again, our brains can make mistakes. Our brains can see different things. They're super cool and talented, but they do have their limitations. Also, we have another question here. The first one, using only height in the visual field. Describe how an artist would draw a second balloon of the same size to show that it is further away than the one in the picture below. And part B, if trees were going to be added, how could the artist use texture gradient to show that one tree is closer than another? So again, this is a visual perception practice question. So here are the answers. For the first one, they talked about how the second balloon should be lower in the sky, so closer to the horizon than the first balloon. So that's linking into how we can use that depth cue of height in the visual field to communicate distance about where the balloon, is it closer, is it further, by using height in the visual field. And the second part was referring to the idea of texture gradient. So you might have picked up on that. So the closer trees should be more detailed, and the trees that are further away should be like less detailed or have texture to give it that idea of receding into the distance. So again, this is just if somebody was drawing a 2D figure, but if you want to make it appear 3D, these are the different cues and like things we can use to make it appear that way. Awesome, we've made it to memory, topic 3. So you're doing well. Thank you for staying along this far. And again, if you have any questions, feel free to pop them in the chat. And we're going to start having a look at memory. So memory, I think, is like one of the coolest topics that we look at in psychology. How does it exactly work? Who knows? People are very fearfully and wonderfully made, and we are complicated and detailed and intricate, and our memory is just like one illustration of that. It's really, really complicated and like awesome. So that's why we have a multiple different models that we're looking at. So we're looking at the multi-store model of memory. Later we'll look at like the working model of memory, levels of processing model of memory. It's all models of memory. There's like elements and aspects of things we might agree with or disagree with in each, but because it's so abstract, we can't like exactly figure out like exactly what it is that we're looking at, how it exactly works, because it's so complicated. So memories are kind of like, in my understanding, is they're stored in like complex networks of integrated neural connections across the brain. It's not black and white and it's not straightforward. But the first store that we're going to be looking at, which is I think a quite common one, and you're probably familiar with a lot of this terminology, is the multi-store model of memory, proposed by Atkinson and Striffen in 1968. And it describes 3 stores, which is where we get the title, the multi-store model of memory, and they function simultaneously and interact with each other. The first one is our sensory memory, which is a very brief store, and the information collected in sensory memory, that's sensory stimuli, is only transferred to short-term memory if attention is paid. So sensory memory is like a store for incoming fleeting sensory information, and actually prevents us from being overwhelmed by the huge amounts of sensory information that we're constantly being overwhelmed with, because we're constantly being bombarded with like visual and auditory stimuli, and we actually can't take it in all at once. So our sensory memory is kind of like the filter to stop our short-term memory from being overwhelmed. So we actually have 2 types of sensory memory. Iconic sensory memory, which is like, it refers to like our visual sensory memory, and it has a duration, so how long the memory lasts, of about 0.3 seconds. And we think its capacity is unlimited, which is crazy. So to illustrate that, think about like someone who's like waving like a sparkler on like New Year's Eve, and then you briefly see the after image. That's like your visual sensory memory, just holding on to that information for long enough for you to see like the trail of the sparkler. Your echoic sensory memory refers to what we hear. So echo, think like noise or sound. So echoic sensory memory is like auditory sensory memory, and iconic is for visual, so think like eye. But echoic sensory memory is like that brief sensory memory for our auditory, like sounds. And an example of why that's useful is that so we can actually like retain word sounds long enough to comprehend a sentence or a phrase. That has a bit longer of a duration of like 3 to 4 seconds, which, you know, kind of makes sense because we need to be able to like put together like whole words and phrases and sentences. Our next level is our short-term memory, which is probably what you've like heard of a lot. A lot of people think about it in kind of like what's consciously going into your head. So it's that like limited store of actively conscious memory. It's what's going on in your head right now. And if the information is encoded, it's then transferred to long-term memory. Short-term memory, most people kind of say it has a duration of like up to 30 seconds. Some say 12 to 20, some say 12 to 30, 20 to 30. But kind of like the up to 30 seconds is like a safe kind of estimate for the duration of short-term memory. But it is quite variable between individuals and like different studies and that. And most people say it has a capacity of only about 5 to 9 things. you can only hold about 5 to 9 things in your short-term memory at once. And again, going to be different between people. And if we get time, we'll also have a bit of a look at like how we remember and forget. And then also long-term memory, which is virtually limitless in capacity. The information requires retrieval to bring it back into conscious awareness. And long-term memory is like where information is like encoded by its meaning or semantically and stored in like semantic networks, which is kind of like think about it like a mind map where interrelated information is like connections to it and things are kind of like encoded according to their meaning. So again, it's kind of like a network of memories in our brain. I kind of think about it that way. So kind of like the main key words, like the memory processes here is like encoding, which is kind of like putting it into a form we can store it. Storage is when we kind of tuck it away for later, and then retrieval is when we bring it back from storage into conscious awareness. So memory kind of passes like from sensory memory. If we pay attention, we can bring it into our consciously aware short-term memory. And then if we like encode it or store it away, we can store it away in long-term memory, which has a virtually limitless duration and capacity. But sometimes we do forget things, and there's a few theories as to why we forget things. But that's our multi-store model of memory. The next thing we can look at is what are actually the types of long-term memory, because there are like different types that we look at. The first is explicit. So that's, for example, like facts, names, images, events. These are like declarative memories. They require conscious retrieval and they're the responsibility of the hippocampus. So how I remember that is I think like explicit, things you need to explicitly think about. And they're like declarative memories. So things you need to declare, like again, facts, names, numbers, and there's 2 subtypes of our explicit kind of long-term memory. There's like the episodic and semantic. So episodic, I think about it as like they're like the personal memories, like episodes from your life. So episodic memories are like memories of events and experiences from your life, and it can be past like or future. And then semantic are kind of like the more like facts, worldly knowledge, general knowledge, things you need to memorize and recall kind of thing. Whereas our implicit memory, think of it more like, internal, a little bit more automatic, is that memories of skills, emotions, preferences, and dispositions. And it's also called like procedural or like non-declarative memories. So procedural, like the memory of like how to do things. So for example, if you are very fluent in playing the guitar, it's going to be quite like effortless for you to play that because it's really easy to recall. It's very cemented in your brain. It's very like implicit. And yeah, they are processed in like the amygdala and possibly also the cerebellum. So again, with my guitar playing analogy, it requires little conscious awareness. It rarely fades with time, except for with disease, and the information is remembered unconsciously and effortlessly. Or for example, singing along to music. It's something we seem to do so automatically. And again, linking in with that idea of like, it's like a procedural memory, which is memory for how to perform certain tasks, skills, actions, or even like conditioned responses. So for example, again, riding your bike, brushing your teeth, these memories are very resistant to forgetting. They're very easy to recall. And with these implicit memories, you often don't have to actually think about them. Like even with, when you're riding your bike, you probably don't consciously think, okay, so how do I ride my bike again? It just kind of very naturally comes back to you. Our next kind of memory model is the levels of processing model of memory. So this model suggests that memory does not comprise any specific number of separate memory stores. Instead, it kind of sees memory as like a continuous dimension in which memories are encoded related to the ease with which they can be retrieved. So the deeper you process the information, and like the more you encode it and the more you connect it with prior knowledge, like the greater chance that you'll be able to retrieve it. So it's all about like the level of processing. which again is like linked to like the number of associations between new and prior knowledge, because the more links you make, the deeper we encode it and the easier it is to retrieve, which is very important when it comes to studying, because there's something just like is a fleeting thought through your mind and you don't think about it again. That's not a very deep level of processing. You're not paying a lot of attention, you're not doing a lot of thinking, so you're probably not really going to be able to recall that or retrieve it because you haven't like encoded it in your brain. Whereas however, if like you're trying to consciously learn something and you're making lots of connections, there's a much greater chance that you're going to be able to retrieve it. So with the levels of processing, again, we can have like those more shallow levels of processing, and we can also have like deeper processing. So the more shallow levels of processing, the first one is like structural processing. So that's when you kind of like remember the physical qualities or features of the words. So for example, is it a capital letter? Is it a lowercase letter? This isn't a very meaningful way to remember stuff, so it's a fairly shallow level of processing. When you have phonemic processing, like that's like when you're remembering like the sounds of the words, which again, not very deep because you're only remembering the sound. You're not giving it meaning. It's always going to be deeper if you give meaning to it, which brings us to our deep level of processing, which is semantic processing. And that's when you kind of like encode or store away memories by like kind of storing it according to meaning. So that's why like kind of organizing things in a mind map is like so helpful, because you're actually creating semantic connections in your brain that's like making connections, and because you're encoding it with meaning, it makes so much more sense and it's so much more likely that you're going to be able to actually recall that information. And there are different ways that we can kind of like make the most of each of these. So for example, with like if you're trying to keep something in your mind, a really shallow way to do that is called maintenance rehearsal. So that's just like repeat and repeat and repeat. So if, for example, I'm trying to remember a shopping list, I'm actually thinking, okay, eggs and bread, eggs and bread, eggs and bread. All I'm doing, that's maintenance rehearsal. I'm just repeating it again and again to keep it in my mind. That's not like a very meaningful way to do it. Whereas if you want to actually deeply process something and encode it in your brain, you're more likely to want to do elaborative rehearsal, which is where you actually give meaning to it and try and intentionally engage in that semantic, deep and meaningful processing. So the underlying message here is that memory grows stronger the more you elaborate on it and the more you link it to things we know and care about. So another model of memory, the working model of memory. So this kind of sees like, this kind of likens short-term memory to working memory. So you can kind of think of them as a little bit similar in this model. So working memory. What is working memory? So, well, first of all, the working model of memory kind of sees memory as made up of these 4 separate but interdependent parts. And so the main one, the kind of like how we define working memory in this model is it's like the store for any mental work that's occurring at any given time. That includes like retrieving information, problem solving, thinking, comprehending sounds, visions. It draws on information from sensory and long-term memory. So working memory is pretty much the information that we're consciously working on. So I kind of think of working memory as like, if you've seen the movie Inside Out, I think of it as like the headquarters. It's what's actively consciously going on. It's what's happening in your brain. So these 4 elements, these are like the 4 separate but interdependent aspects of working memory. So this is the working model of memory. And so these are like kind of like the 4 elements that we would consider to be part of working memory. The first one is the phonological loop. Think phone, sounds, hearing. The phonological loop is our auditory working memory. It's the storage of what we hear, and it's what helps us to understand sentences. And then we also have a second one is the visuospatial sketchpad, which is the storage for visual short-term memory. It's what we see. It's also what helps us to like visualize if we move things and change things. It's our visual working memory, what we're kind of like visualizing in our mind. Third one is the central executive, and that kind of puts together like the sound and the visual memory, and it also controls our attention, which is what enables us to mentally manipulate things. So the central executive, I see it like the control center, kind of like the boss, kind of like the control panel up the front of the brain in the movie Inside Out. It has like three main functions, which are inhibition, which is screening out irrelevant information. So I'm going to choose not to pay attention to that. Switching, which is when you choose to like change your focus to something else. And then also updating, so modifying items brought in from long-term memory before returning them to long-term memory through the episodic buffer. So I kind of think about that as like the memory tubes in Inside Out. So I kind of, I think about it like the little memory ball comes in from the tube. If they have to like modify the memory or like make changes to it, or if they like add to the memory, and then they can send it back to the memory tubes. And then we have the episodic buffer, which I kind of think about like a conveyor belt, like the memory tubes in the movie. So it's how we like retrieve information from long-term memory. And then by doing that, you can like make associations with this memory from long-term memory, and then you can add information to it in working memory, like in the control center in Inside Out, and then you can like select and encode information and then send it back to long-term memory. So that's our working model of memory, all about like the conscious what's going in my brain right now at this moment. Memory, the amygdala and hippocampus are quite involved in that. I definitely encourage you to do some extra research on that. A few brief things like the amygdala, it teaches you to detect danger and become afraid. It enables acquisition and use of classically conditioned memories. There's high levels of noradrenaline in the amygdala, which lead to stronger memories. It's activated by strong emotions at the time of encoding, and the emotional reaction stimulates adrenaline release, which is associated with like the fight, flight, freeze response. So when we think about the role of the amygdala in memories, I definitely associate that with like our emotional association with memories. So when you have a memory and it's like very associated with an emotion, that definitely makes me think of like the amygdala's role in memory. The hippocampus is crucial in consolidation of new declarative memories, which we talked about just before, as well as the transfer of memories to the cerebral cortex for storage. The hippocampus has a really, really big role in memory. You definitely need to do some research into that as well. I think it also has a role in spatial memories, and it has place cells, which help us to create like a mental map. It also might help us like retrieve memories, not as important for procedural memories. And it does work with the amygdala so that declarative aspects of emotional events are remembered. So to kind of like summarize the main difference, amygdala is kind of like the emotional aspect of memories, and the hippocampus is kind of like crucial in the consolidation of new declarative memory. So remember, declarative memory is like the facts, the events, the things you need to consciously recall. So hippocampus has a lot to do with like transfer of memories and the storage of the memories. The cerebellum is another really cool one in memory, which is like that little kind of like mini brain at the base of the brain near the brainstem. And the cerebellum encodes and stores like kind of like conditioned reflexes, like automatic movement is kind of how I think about it. Like it encodes and temporarily stores like procedural memories. So again, we're thinking hippocampus, declarative memories. Cerebellum is where we're kind of getting more into those procedural memories. It can also kind of like read the mental map created by the hippocampus, and it also has a major role in like coordination and motor learning. So how I kind of think of the cerebellum is it's about like remembering how to perform a motor skill. It's like a procedural memory store, and each time you practice that new skill, it'll add to your procedural knowledge of the task. And that's like why new things are harder as well, because you're putting so much conscious effort into like learning this new skill, but as you practice, it gets more smoother and automatic. Because the cerebellum has a lot to do with like voluntary movement as well, like that balance and coordination. But as far as memory goes, it's more about like that coordination and remembering how to do a movement smoothly. So for example, I've been learning to play the ukulele recently. I've been really enjoying that. And when I first started, it was like so hard because I'd never played a stringed instrument before. I didn't know where or how to put my fingers, and it was just so challenging for me. But as I practiced that and consolidated my procedural memory of that task, it got so much easier with time. So much easier with time. And then it just, started to come back to me more naturally because of that procedural memory of that task, which again becomes more smoother and automatic with time. So the cerebral cortex is also involved in memory. A lot of people see it as like the storage site of declarative memories in long-term memory. So episodic memories are kind of like stored throughout, but especially in like the right prefrontal cortex. And again, this is all quite specific information, so don't be overwhelmed if you can't remember all of this all at once. Semantic memories are stored throughout the brain, but especially in like the temporal and the frontal lobes. And then pieces of memory are usually stored in an area or the lobe where the information was initially processed in. And again, it's very important in reconstruction, especially for declarative memory. But again, my main takeaway for you guys about this slide is all mostly just like about making sure that you're really aware of the idea. The memory is incredibly complicated. Like there are so many different like elements of like the brain that like play a role in it. And it is very complicated, and all of the different elements of the brain work together to make that happen. There are also different like types of retrieval. The three main types are recall, recognition, and relearning. So in recall is when we're retrieving information, but all you need to do is identify the correct option without being given a list of possible answers. And then in recognition, that's when you need to only select the correct option from a list of alternatives. But relearning is where you've kind of forgot what you've learned and you actually need to relearn it all over again. But it is a lot quicker than learning like the first time around. Like it's a much faster process. You make much less errors, and it's just generally an overall like a much smoother process. So we have these three types of like retrieval. Recall, where you need to bring the information back with like a very minimal cueing. Recognition, so recall, I kind of think about it like our recall, I'd say, is kind of like our like short answer questions on a test, I guess. Whereas recognition is like our multiple choice. All you need to be able to do is select the correct response from a list of alternatives. Whereas relearning is like when you got to learn it all over again. But again, it is like much quicker and more efficient than like learning something for the first time. And we are running a little bit low on time. We only have a few moments left. So to finish this up, I'm just going to quickly touch on the concept of forgetting long-term memories. So how do we actually forget? There are actually a few different theories on this, and it is quite interesting. The first kind of theory is the idea of ineffective coding, which is kind of known as encoding failure theory, which is kind of the idea that maybe the information was never actually encoded or stored properly in the first place. And we can kind of contrast that with ineffective retrieval, which is referred to as like retrieval failure theory, which is quite like given away in the name, where we actually are just failing to retrieve it. So that's the idea that the memory is in there somewhere, like it was encoded, it is in there somewhere in the back of our mind, but we're just failing to retrieve it. So that's where the name comes in really handy with that retrieval failure theory, is the idea that yes, the memory is there, but we're just struggling to retrieve it. And the idea of that is that there's like poor retrieval cues. So cues are kind of like things that like prompt us to remember our memories. The kind of two main types that we look at are like context-dependent cues, which are like cues in the external environment, like a sound or a smell. And then we can also have like state-dependent cues, which refer to the internal environment of the learner when the learning took place. And it includes like psychological and the physiological state of the learner, such as like their mood and everything. And another really key thing about that is if we're trying to learn information if we're trying to recall information under similar conditions to which we originally learnt it, we're actually going to be much more likely to retrieve it. And again, that brings us back to the importance of like encoding information properly, like encoding it according to meaning and semantically, so that you can actually encode it and intentionally, if you really want to remember something, actually intentionally placing the right cues so that you can retrieve that memory. And there also are multiple ways that we can try and improve our memory and reduce the likelihood of forgetting, like mnemonics, the SQ4R method, which we have here, and a whole lot of other ways. But we actually have run out of time for those for today, so we won't quite get to those. But again, remember, the slides are available for you to download, so if you would like to go through this afterwards, you are more than welcome to. So yes, I'm sorry we didn't quite get through all of our content today, but again, if you would like to go through the slides, you are more than welcome to, and hopefully that'll be a really valuable resource for you as your studies and as you prepare for, I guess, the year ahead at school. But I just wanted to say a massive thank you for joining us today. I really appreciate everyone that jumped on, and I hope this lecture was actually able to be really, really helpful for you all, and you were able to get some good tips and tricks and knowledge for it, and that you're all feeling really comfortable and confident about year 12. I'm sure you will do really well in your studies. And thank you again for joining us, and I hope you all have a really good rest of your week. So thank you all so much again, and hopefully I'll see you around in some of the ATAR notes, lectures or resources in the future. So thank you so much again for your time. Have a great rest of your day. Thanks, guys.